Okay, guys, so we're going to start our presentation. Everyone seeing the screen, right? Yes? Everyone can hear? Okay, yes. All right, so we can start. So our presentation today is on microorganisms and medicine. And I think you guys should be already familiar since we already did a lecture on aseptic techniques, so you guys know the importance of what microorganisms are. We're just going to go over what they are quickly. So what microbiology is the study of microscopic organisms such as bacteria, viruses, archaea, fungi, and protozoa. Microorganisms are minute living things that cannot be seen with the naked eye. So parts of microbiology. So microbiology is divided into different subsections or headings, and these are bacteriology, which is the study of bacteria, mycology, the study of fungi, phycology, the study of algae, protozoology, which is the study of protozoa, virology, study of viruses, and parasitology, which is the study of parasites. So these are the different microorganisms, or some of them not necessarily microorganisms, some of them could be considered macro, but these are the different subheadings or parts of microbiology. So this is a little cartoon that I made for you guys to see. So right here at the right hand, it says, you might call them scientists, but for me, they are paparazzi. And then you have the one at the bottom saying, I'm fed up with this guy, let's become pathogenic. So, all right, so we're going to continue, guys. So the history of microbiology. So illnesses and disease were actually thought to have a supernatural cause. So many people thought that if you were sick, it was due to like evil spirits or the wrath of God. So persons who had flu or cholera, they thought that, oh, it's because they sinned or they did something bad in their lifetime. That's why they had certain diseases. And the possibility of illness and disease being linked to unseen microorganisms was not thought about or postulated widely until the invention of the microscope and a series of other experiments that we'll be going into. So we're going to be talking for the first part about the history of microbiology and how we came across the different experiments and how we, how we be able to know what we know now in microbiology. So the miasmatic theory, this was actually came about by Galen of Pergamon. And Galen of Pergamon, he suggested that disease such as cholera, chlamydia, or the Black Death were caused by miasma or bad air. And so the theory basically held that these epidemics were caused by miasma arising from decaying organic matter. Although incorrect, the miasmatic theory spurred the development of better sanitation and hygiene practices. Aqueducts were built and brought in fresh water. Sewers were built that carried away waste and sewage, and this practice protected the Romans from many waterborne diseases. Okay, so the theory of spontaneous generation. So this theory existed to explain the origin of some forms of life. So Aristotle, he proposed that life arose from non-living material if the material contained pneuma, which is like some form of vital heat that you would find in the air. As evidence, he noted several instances of appearance of animals from environments previously devoid of such animals, such as fish appearing in puddles, even maggots being found on food, flies forming out of feces. But this is before the invention of microscopes. So what they didn't know was, when these maggots were formed on these food, it was actually flies that laid their eggs that were too small for human eyes to be seen. So also these things like frogs or animals being born in lakes and finding new species. It wasn't spontaneous generation, but just us not being able to see these species or different creatures because we didn't have the tools back then to see them, but now we do. So Francis already, he came up with an experiment to debunk the idea of spontaneous generation. He believed that things just didn't pop out of the air just like that. So he had an experiment where he had 6 jars of meat where he placed in the jars, right? 2 of them were open, 2 of them had like gauze over them, and 2 of them were tightly sealed with the corks. And what he observed is that the 2 that he left open, 2 containers that he left open with the meat, that he saw the formation of maggots on the meat, and the 4 other containers, which had the 2 cork seal container and the 2 paws, there was no formation of maggots. So he basically hypothesized that since there was no formation of the maggots, it had to be due to something from he was saying that basically the flies must have laid eggs that we can't see, and those eggs hatched to form the maggots. And that's how the theory of spontaneous development generation was actually debunked. So I think you guys have been already familiar with it. In the early development of microbiology, you know, we had somebody called Anton von Leeuwenhoek, and he coined the existence of the use of microscope by observing pond water. He discovered microbes which he called animalcules. And this was actually the first time in history in which persons were aware of the existence of bacteria, because he was able to document these bacteria and he sent them to the scientific group of microbiologists, and that's how they were able to say, okay, bacteria actually exist. They're actually small microscopic organisms that we can't actually see. So everyone's following, right? Everyone's good? Okay. All right, so next we have the next person, who is Louis Pasteur, and he performed experiments again to also disprove the theory of spontaneous generation. Okay. So he actually filtered air through a cotton plug and placed the plug in an infusion broth, which is, the infusion broth is basically the like a medium in which we use to grow up the microorganisms. So this medium would have like different things that they're needed to grow, like protein, fats, just anything that they would need to be able to grow and thrive in that certain environment. And the broth became cloudy present in the air. And then he placed boiled infusion broth in the swan necks, and then the flask remains in sterile unless tilted or net broken. So this is an sorry, let's go to the next page. So this is an example of the experiment he did. So what Louis Pasteur did to actually disprove the spontaneous generation theory is that he actually boiled broth, which I was saying is the medium in which we use to grow up these microbes, to get rid of any microbes, that unwanted microbes that may have been present. So that we don't, because he's trying to disprove that if you heat these microorganisms to a certain temperature, they would die out, because their enzymes would no longer be at an active level and they would die. So when the swan neck is intact, allowing air, what happens is the swan neck is made up of cotton, right? And it allows air to come in, but at the same time, it traps certain microorganisms. So what he did was he boiled the broth tube, and then he allowed air to come in, but it would also trap microbes at the same time. So no microbes are allowed to enter. He waited 3 days, and then he tested it, and there were no microbes. But when he boiled the broth tube to get rid of the microbes, and then he removed the swan neck, allowing both air and airborne microorganisms to enter, he found that 3 days later, it actually contains microbes. So this was actually disproving the whole theory of spontaneous generation, saying that, like living matter can just appear out of non-living matter. So that is what the spontaneous generation theory is basically saying. And he debunked that theory, saying that cannot happen. So development of early microbiology. So Edward Jenner, he was actually came up with the first vaccine in 1796 for smallpox. So Edward Jenner, he observed that milkmaids who had been previously infected with a similar disease called cowpox, which like you would have lesions on they would have lesions on their hands, which they would get from like milking the teats of the cows. So he observed that, and the principle behind the vaccination is that when these milkmaids were infected with this more milder form of disease, if they were able to contract or get smallpox, they would not be affected because their immune system would have already been accustomed, or it would have had some form of exposure to this form of agent already, so it's able to fight off the disease. So Joseph Lister, he introduced the antiseptic technique, which is the use of a phenol as a disinfectant. And actually, he's the person who came up with using Listerine. So Listerine is actually named after Joseph Lister. Alexander Fleming, he actually discovered the first antibiotic, which is called penicillin, and it is produced by a mold that contaminated bacterial culture plate. And it actually led to the discovery of other fungi that secreted antibacterial substances. Okay, so this is the phylogenetic tree of life, It's, and you know, this is separated into your bacteria, archaea, eukarya. All right, and we also have, it's not actually classified under this because, but also you have viruses, but viruses is kind of like its own thing, so it's not really classified under because of different, because you know viruses are different from bacteria in different ways, and that viruses don't, they need like a host machinery to replicate. So you know how bacteria, they're able to just grow and replicate on their own. That's not the case for viruses. So we're going to be focusing on talking on mostly just bacteria and viruses for this. So also classification. So the classification of microbial diversity. Classification is actually the organization of organisms into progressively more inclusive groups on the basis of either phenotypic similarity or evolutionary relationship. This is done through systematics and the study of diversity of organisms and their relationships. And taxonomy is actually the science in which these organisms are characterized, named, and classified according to several defined criteria. So microbiology and medicine. So medicine microbiology is actually a large subset of microbiology, and that is actually applied to medicine and is a branch of medical science, which is concerned with the prevention, the diagnosis, and the treatment of infectious diseases. In addition, this field of science studies various clinical applications of microbes for the improvement of health, and there are 4 kinds of microorganisms that cause infectious disease, as we said before: bacteria, fungi, parasites, and viruses, and 1 type of infectious protein, which is also called a prion. So for this, we're going to actually be talking about bacteria and viruses and how we actually classify them. And we're going to actually go into a little bit of talking about vaccines and antibiotics as well. So bacteria, so these are single cell organisms, and they tend to lack like a membrane bound organelle. So these are like, hold on, somebody said something in the group. Okay, wait, hold on, guys. Somebody's in the break room. I think somebody got connection. Okay, all right, we're good. All right, so bacteria, these are single cell organisms that divide by binary fission, and they tend to lack membrane bound organisms. They tend to be small in size, and they actually, so these membrane bound organelles tend to be like the Golgi apparatus, or just some of them don't even have like a real nucleus. It's actually very simple, so they have to have very simple organelles in them. They just have like the basic stuff that is needed. So we classify bacteria in many different ways, but the 3 ones that we'll be focusing on today are the shape, how they're able to retain certain dyes, and the ability to grow with or without air. So bacteria are also capable of independent replication, unlike viruses, which actually need a host, such as an animal or a plant or humans, in order to grow and reproduce. They cause most of infections seen in the hospital, such as pneumonia, bacterial meningitis, cellulitis, and they're treated with antibiotics. So this is actually the normal flora or microbiota. I think I mentioned this to you guys before. So these are microorganisms that naturally colonize the body but do not produce diseases under normal conditions. So you have your staphylococcus, which is found in your upper respiratory tract, your streptococcus, your haemophilus, and then on your skin you actually have your staphylococcus. Staphylococcus is one of your normal microflora, as it said, but it can cause hospital diseases as well. So you can probably get like meningitis or different diseases that you can get if you're found that your immune system or if you're immunocompromised when you're like in the hospital, you would more likely get from staphylococcus. Gastrointestinal tract, you have any aerobic bacteria or even facultative anaerobic bacteria such as Escherichia coli that you'll find, Streptococcus again, and Candida species. Genital tract, Lactobacillus, Streptococcus species. So these are on your normal microbiota or flora, microorganisms that will normally be found on your skin or even parts inside of your body as well. So bacteria classification and identification, and so this is morphology, also the same as shape. So there are three basic shapes of bacteria, which is your spear and crocodile. So these are round or oval shaped bacteria measuring about 0.5 to 1 micrometer. So just for you guys to know, this right here means micro. So this is like a micrometer. So all right, so in diameter, when multiplying cocci may form pairs, chains, or irregular groups such as, so example, your staphylococci is actually considered a cocci-shaped bacteria. So rods, these are stick-like bacteria with rounded, tapered, square, or swollen ends. They measure about 1 to 10 micrometer in length by 0.3 to 1 micrometer in width. An example of this is your E. coli. The spiral-shaped bacteria, these are small, regular, coiled, rigid organisms. They measure about 3 to 4 micrometer in length, and each coil measures about 1 micrometer. So spirula or spirulum are motile groups of flagella at both ends are an example of this group of bacteria. So these are examples of the shapes I was telling you about. So I was saying that some of the spheres or cocci, they can have them being diplo, they can have them being tetra, they can be actually single as well. So your staphylococci tend to be grouped together right here. They tend to form clusters right here. So these are your staphylococci. They tend to form clusters. With diplococci, they come together as two forms, your tetrads. Then you have your rods such as bacillus, and then Clostridium botulinum. You have your spirals such as spirulium, spirochetes. You have your vibrios. So these are just examples, guys. Let me just erase that. All right. Okay, so gram staining. So this is a technique in which we use to stain different organisms. So we have usually our bacteria, we classify them according to gram positive or gram negative, right? And I will go into that soon, right? So the staining technique is, the first part of the staining technique is when we stain them, we stain both our gram positive and our gram negatives with a crystal void, right? And then we'll wash it off. So we usually use a crystal ball to stay for probably like 10 to 30 seconds. Then we'll wash it off with like a mordant or what we call, or the iodine. So this helps to fix the dye in place is towards the cell. And then we'll use a decolorizing agent or an alcohol. And this actually washes out the alcohol, this is actually washes out the dye, the crystal violet, and then the counter stain, which is the safrarin. So what happens is that gram negative and gram positive, they actually have something which is called, okay, so I'm going to come back to this page, hold on. So this is actually a picture of on your left hand side you have the gram positive, and on your right hand side you have your gram negative. So you guys don't need to remember all of this. The only thing I want you guys to note is that your gram positives actually have, let me just annotate so you guys can see what I'm, so your gram positives actually have like a thicker peptidoglycan layer, and your gram negatives, their peptidoglycan layer is actually very thin. So this is their peptidoglycan layer is actually very thin compared to the peptidoglycan layer of the gram positives, right? Okay, so we're going to go back now. Sorry. Why isn't it coming out? Oh, sure, let's remember this for this laptop. All right, sorry. So because the peptidoglycan layer of the gram positive bacteria is more thicker than the gram negative, they're able to hold on to the crystal violet dye more or than the gram negative. So when the dye is placed into, when they're colored with the dye, it becomes fixed into their peptidoglycan layer. It helps to hold the dye properly. So when the decolorizing agent or the alcohol is added, it's not removed compared to the gram negative. So as I said, it's a technique used to differentiate two large groups of bacteria based on their cell wall constituents, because they actually have a higher peptidoglycan layer. And the gram stain procedure distinguishes the gram positive from the gram negative by coloring these cells red or violet. So gram negatives at the end of the experiment, they'll show as like a red or pinkish color, and then the gram positives then will show as purple. All right, and as I was saying before, see that the gram positives, as you're seeing right here, actually have a thicker peptidoglycan layer. So that's how they're able to hold the crystal violet dye, and the gram negative actually have a smaller peptidoglycan layer. Okay, so oxygen requirements. So we have two main types of oxygen requirements when it comes to microorganisms: the ones that are anaerobes and the ones that are called aerobes. So your anaerobes are the bacteria that only grow in the absence of bacteria of air. Sorry, I don't know why I have that there. Bacteria that only grow in the absence of air, sorry, such as Clostridium. And then the aerobes are bacteria that require oxygen to grow, such as Bacillus subtilis. So when you have them in your culture medium or tubes, you'll find that the ones who are aerobes, who like oxygen, will grow at the top of the tube. From your tube C to the aerobes, like, so they'll tend to be found like at the top of the tube right here, and then your obligate anaerobes will tend to be found at the bottom of the tube right here. But you do have some organisms that are not completely aerobic or completely anaerobic. So you have like facultative. So facultative just means that they anaerobe. So facultative anaerobes, such as E. coli, which I mentioned before, they're able to grow at like small amounts of oxygen, but they grow better without oxygen as well. And then you have even like stuff like microaerials, which are able to grow like at small amounts of oxygen. So they'll probably found like somewhere around here of the tube. But I don't want you guys don't need to know that, I'll just mention it. So just you guys need to just know the obligate aerobes and your obligate anaerobes. So the aerobes, as I said, they're the ones that require oxygen to grow, and the anaerobes don't require any oxygen to grow. They prefer to grow without any oxygen, such as Clostridium. All right, so we're going into this. So viruses, they are small, about 0.02 to 0.35 micrometer, and most are actually below the resolution of the light microscope. These infection agents, hold on, give me a moment, guys. Okay, sorry about that. So as I was saying, viruses are actually small, 0.2, 0.02 to 0.35 micrometer in size. So they're actually below the limit of the resolution of our light microscope. So most times you'll actually have to use different microscope, maybe like an electron microscope in order to view them. Infectious agents with both living and non-living characteristics. So as I was saying, they are living characteristics that they reproduce at a fantastic rate, but they lack the ability to self-replicate outside of a host cell. So they're considered to be obligate intracellular parasites. So parasites are basically organisms that use, like, they're attached to like other organisms which you call the host in order to grow. So they can't grow on their own, and that's basically what viruses are. They're parasites. They're made up of organic molecules. They're able to mutate. So even like a lot of persons are saying that, the COVID-19, it may actually be like a mutated virus, because so that's what a lot of people are saying. Most viruses possess either DNA or RNA, not both. And viruses, so it's non-living characters, are they acellular, contains no cytoplasm or cellular organelle. They carry out no metabolism on their own and must use a machinery to replicate. So how to classify viruses? So we classify viruses based on their genetic material. So do they have DNA? So their DNA viruses are the ones that contain DNA as their genetic material. RNA viruses contain RNA as their genetic material. And morphology, are they helical? So do they have the capsomers bonded, which give them the spiral shape? They have the polyhedral, and these tend to be flat. Spherical, which are round, and then you have your more complex shapes. So some of them may actually have like membranous envelopes surrounding the capsid, and then you have the type of host that they may attack, such as bacteria, animal, or human. So these are the different viral shapes I was talking about. So you have the polyhedra, which is an example of an adenovirus, the helical, the tobacco mosaic virus, the spherical, which is the influenza, and then you have more complex viruses, which is your bacteriophage. So now we're going to just go into antibiotics and vaccines. And so antibiotics are the medications that destroy or slow down the growth of bacteria. They include a range of powerful drugs and are used to treat diseases such as bacteria. As we were saying, antibiotics cannot treat viral infections such as cold, flu, and most cough. So, your penicillin, your tetracyclines, aminoglycosides, and your clindamycins are examples of antibiotics. And then you have your vaccines, which is a biological preparation that provides active acquired immunity to a particular infectious host. Vaccine typically contains the agent that resembles disease. So it may be a weakened form of the disease that the vaccine may be carrying, or it may actually be the killed form of the microbe or its toxins. The agent actually stimulates the body's system to recognize the agent as a threat and destroy it, and further recognize, destroy any microorganism. So maybe sometimes if you ever got a vaccine when you're probably like in prep school, you might have remembered like shortly after getting the vaccine, you might have had a fever, you might have been like slightly sick. So what the vaccine does is exposes your body to the weakened form or the killed form of the organism. So if you're ever supposed to contract or get the disease, your body is able to fight it off because it was already pre-exposed to it, so it's able to produce antibodies and such to fight off this disease. Okay, so this is the end of the presentation. So